For decades, the pursuit of a single cause for autism spectrum disorder drove researchers to look for one specific flaw. They searched for a singular autism gene or an isolated environmental trigger that could explain the condition. That search proved to be a scientific impossibility. The very concept of a spectrum reflects an immense range of presentations. We see profiles involving profound intellectual disability and nonverbal communication existing alongside highly verbal, specialized cognitive profiles. Because of this sweeping biological diversity, the contemporary medical consensus has moved entirely away from the idea that one isolated factor produces the disorder. Major global public health and medical institutions, including the CDC, the World Health Organization, and the National Institutes of Health, now uphold a completely different framework. They recognize autism as a polyetiological condition, the result of highly intricate interactions among genetic predispositions, epigenetic modifications, and environmental exposures occurring during the most vulnerable windows of brain development. To understand how this works, we have to trace the biological sequence chronologically, starting at the very beginning with germline DNA and following the developmental chain reaction all the way to the macroscopic wiring of the brain. We must abandon simplistic explanations. The autistic brain is the endpoint of a highly orchestrated, multifactorial process. The foundation of this process lies in a genetic architecture characterized by extreme heterogeneity. More than 1,000 distinct genes distributed across various chromosomes are actively implicated in autism susceptibility. To navigate this volume of data, the scientific community relies on the SFARI gene database, a centralized registry that utilizes extensive clinical and transcriptomic data to evaluate exactly how causative specific genes actually are. This data matrix illustrates the SFARI scoring system, assigning confidence levels based on peer-reviewed data. The top tier, Category 1, represents genes clearly implicated in autism. Reaching this requires a strict false discovery rate under 0.1 and 3 reported de novo mutations. Those de novo events are spontaneous mutations, completely absent in the parent's somatic cells. This contrasts with inherited variations, where familial genetic reservoirs are passed down generationally. The risk of these mutations increases with parental age, but mechanisms differ. A woman's oocytes age alongside her, increasing the likelihood of chromosomal non-disjunction. Conversely, paternal stem cells replicate constantly, driving a cumulative accumulation of spontaneous point mutations targeting brain development. Yet, many individuals carry these exact genetic variants without displaying any clinical symptoms. The genotype alone isn't destiny. It requires an environmental or epigenetic catalyst to trigger the presentation. Those inherited genetic anomalies ultimately dictate exactly how the physical tissue of the cerebral cortex is constructed during early embryonic development. To observe these developmental errors, scientists reprogram patient skin cells into human brain organoids. These models grow into tissue clusters that recreate prenatal cellular alterations. These organoids reveal a consistent abnormality, an overproliferation of excitatory cortical neurons in the forebrain, creating a stark cellular imbalance before the brain is even fully formed. Once generated, young neurons must migrate outward to form the distinct layers of the cerebral cortex, a highly sensitive process known as cortical lamination. This cross-section shows where the migration fails in the autistic brain. While healthy green neurons reach the surface, the red ectopic neurons stall or divert, becoming misplaced in the lower cortical layers. Because these ectopic neurons fail to reach their destinations, they suffer a delayed switch from excitatory to inhibitory signaling, maintaining immature, hyperexcitable characteristics. These early cascading errors establish a permanent physical imbalance between excitatory signaling and inhibitory control, long before birth. Once neuronal migration sets the physical structure, the brain's functional capacity is determined by how those neurons connect, a process called synaptogenesis, and how those connections are subsequently refined. In typical development, the brain produces an excess of connections, then undergoes synaptic pruning to eliminate weak or redundant synapses, creating high-signal, low-noise neural networks. This network model highlights the divergence. On the left, a neurotypical network prunes down to clear pathways. On the right, the autistic network fails to adequately prune, remaining hyperdense, chaotic, and neurologically noisy. This is no longer theoretical. Groundbreaking imaging at the Yale Child Study Center allows researchers to measure this directly in living humans using PET scans. The research revealed that autistic adults possess a staggering 17% lower synaptic density across the entire brain compared to neurotypical individuals. This lower density correlates directly with clinical behavior. Individuals with fewer synapses exhibit profound traits, including severe sensory issues and difficulty interpreting social cues. This translates to macroscopic wiring differences. fMRI data from infants shows abnormally strong connections between the brain's salience network and sensory motor regions. With inadequate pruning and hyperconnectivity to sensory areas, the autistic brain is wired from birth to prioritize localized physical stimuli over complex social interactions. Since static DNA sequences cannot fully explain these outcomes, researchers look to epigenetics, biochemical mechanisms like DNA methylation that dictate whether a genetic vulnerability is actually transcribed and expressed. One of the most critical environmental modifiers is maternal immune activation. When a pregnant mother experiences a systemic viral or bacterial infection, her immune response releases a flood of pro-inflammatory cytokines. Specific cytokines, such as interleukin-6 and interleukin-17, are capable of crossing the placental barrier and entering the highly sensitive fetal compartment. This biochemical view shows how the exposure causes epigenetic remodeling. As a cytokine molecule attaches to a neural receptor, it forces a methyl group to tightly lock onto the DNA strand, chemically silencing the expression of vital synaptic genes. Beyond maternal health, external toxicants act as epigenetic triggers. Widespread exposure to ambient air pollution, specifically fine particulate matter like PM2.5, significantly increases neurodevelopmental risk. The impact of these pollutants depends entirely on the fetus's unique biology. The GSTM1 gene, for example, regulates the body's ability to clear oxidative stress. If a developing fetus possesses a mutated variant of this detoxification gene, it simply cannot filter the neurotoxins. Ubiquitous environmental stressors do not cause autism in isolation. They inflict permanent cytoarchitectural damage, specifically when they exploit a pre-existing genetic vulnerability. To accurately define this biological reality, the medical community must actively dismantle the pervasive, unscientific myths that continue to misdirect public health efforts. The most damaging myth originated in 1998 when a fraudulent paper claimed a link between the MMR vaccine and autism based on completely falsified data. Epidemiological research has provided a definitive statistical counterproof. A massive study analyzing the health records of nearly 100,000 children demonstrated that the MMR vaccine does not increase the risk of autism, even in the most genetically vulnerable populations. Similar statistical misinterpretations drive modern controversies, such as recent FDA discussions regarding a possible association between maternal use of acetaminophen during pregnancy and neurodevelopmental disorders. This flowchart illustrates confounding by indication. Maternal fever leads to two outcomes: acetaminophen use and an elevated autism risk triggered by inflammatory cytokines. The underlying fever is the actual biological threat causing neurodevelopmental distress. The medication is simply a statistical proxy. Misattributing the cause of the disorder to safe medical interventions actively endangers fetal brain development by discouraging pregnant women from treating actual biological threats. Autism spectrum disorder is a condition defined by thousands of interacting biological variables that defy any search for singular triggers. Over 1,000 highly confident genomic variations provide an initial unstable structural blueprint. That blueprint is then physically altered by the epigenetic consequences of the gestational environment, reacting to everything from maternal immune storms to airborne toxicants. The outcome is a physical brain defined by ectopic neuronal migration and deficient synaptic pruning, creating a dense connectome prioritizing local sensory processing. By abandoning historically flawed theories and studying this multifactorial biological reality, the scientific community takes the necessary steps towards identifying accurate biomarkers and providing meaningful support for the neurodivergent brain.